Hello, Grade 10. Now we will talk about the chromosome structure. We have finished in the last video talking about the cell cycle. Let's start now with the chromosome. The chromosome condenses at the start of mitosis. Here you can see the double helix of the chromosome. The chromosome, okay, or the genetic material, sorry, I have to say the genetic material inside the cell is in the form of DNA, right? It is in the form of DNA double helix. Each continuous double-stranded DNA molecule make one chromosome. What happens now? This is a cell. The cell contains a nucleus. This is a cell. This is the nucleus. Inside the nucleus, you can see this, what spaghetti-like shape, which is named as chromatin. How does this chromatin formed? The DNA, the genetic material, is warps around a protein called histones, forming the chromatin. So this chromatin consists of what? DNA consists of? This is an important question. The chromatin consists of DNA plus what? Histones. What happened? The cell now wants to divide. It cannot divide its genetic material in a chromatin form. So what will happen? The chromatin will compact and warps around a protein to form a spiral coiled DNA. So again, this spaghetti-like shape, okay, will be converted to be spiral coiled DNA. The spiral coiled DNA will continue or will coils more and more till make a chromosome shape. So here you can see the chromosome. The chromosome inside the cell nucleus, you can see it only when the cell is ready for the division or is dividing actually. So what is the structure of the chromosome? Here you can see the chromosome structure. Okay, I will make it larger for you. Here you can see the chromosome structure. The chromosome consists of the DNA plus protein. DNA plus what? Protein forms the chromatin. And as I told you that the chromatin will condense more and more till form this chromosome shape. One half, do you see this red part? One half of the chromosome is called chromatid. One half is called chromatid. Okay, so this is one half. This is the other half. The other half here is called also chromatid. So the chromosome consists of two sister chromatids. Consists of what? Two sister chromatids. And are attached together by a point called centromere. Okay, so the sister chromatids are held together by or at a point called centromere. Okay, what about these far ends? Here, here, and here. These parts called telomeres. These parts called what? Telomere. They are far ends of each chromatid. They protect DNA and don't include any genes. It helps the chromosome in division, and it protects the chromosome during the division. Also, the telomeres is responsible for aging. Telomeres are responsible for aging. Okay? Okay, so let's start with the interphase. Okay. The interphase, longest phase of the cell cycle. Okay, this is a repeated word, consists of G1S, G2. The cell contains a pair of centrioles. Okay, this is an important to know that to see the cell and to say in which phase does this cell occur. Actually, this cell is occurred in the interphase. How do I know this? When I see the centrioles are free and you can see the nucleus is complete, you can say that this cell is in the interphase. The cell chromosome have duplicated but are in a form of chromatin. So in interphase, the DNA will be in form of what? Will be in a form of chromatin. This is right, okay? I can give you a question. In interphase, the DNA will be in a form of chromosome? No. In interphase, the DNA will be in a form of chromatin. Okay, so after that, let's discuss the mitosis. In mitosis division, it produces two genetically identical daughter cells. This is the parent cell to N. It will produce what? Two identical cells. So when you see this graph or this figure, you will say that this is a mitosis. The mitosis consists of pro, meta, ana, telophase. I remember it as PMAT. PMAT. What is a PMAT? Pro, P for prophase of course, M for metaphase, A for anaphase, and T for telophase. Okay, what happened? Here you can see this cell is, both cells are in interphase. They didn't divide yet. Okay, now here you can see the centrioles are getting far, and the nuclear membrane here is what is cutted. The nuclear membrane is cutted. Can you see this? This is the, if you compared it with the cell above, you can see the nucleus. Here you can see the nucleus as a complete circle, right? But in the prophase, no, the nucleus is cutted, or the nuclear membrane is disappeared. The nuclear membrane is disappeared or began to disappear. This is the first thing that happened in the prophase. The second thing, the chromatin condensed into chromosome. Can you see this? X shape, they are chromosome. If we compare it with the above, no, the genetic material here is in a form of what? Spaghetti-like shape, because this is interphase, but here it is what? X shape. It is in a form of chromosome. Also, you can see that there are something called spindle fiber is formed. Do you see this part? These are spindle fibers. You will know their function later. So this was the prophase. In the metaphase, this is important. The metaphase is very, very important. Do you remember the spindle fibers? The spindle fibers are attached to the centromere. These lines is called, or these lines are called spindle fibers. They are attached to the centromere, okay? And here you can see in the metaphase, you can see all the chromosomes are lined up. So the chromosomes here are lined up in the middle of the cell, okay? In the middle of the cell, okay? After the metaphase, you can see the anaphase. Here you can see that the spindle fiber began to condense or pull the chromosomes or not the chromosome, pull the chromatids toward itself. So again, these lines, these spindle fibers, these lines begin to condense. It will getting shorter. Condense means that it becomes shorter. When it becomes shorter, it pulls the chromatids toward itself. Okay? So spindle fibers condense and separate sister chromatids to opposite sides of the cell. The telophase, which is the last phase in the mitosis division, here the chromosomes begin to uncoil because we want the cell to be as it was, okay, or as in the beginning of in the form of the interphase. Again, so the telophase, it is the last phase in the cell division. Here, the chromosome begin to uncoil, the nuclear membrane reforms, the spindle fiber disappears, and the phase follows the telophase is called cytokinesis. Do you see this construction? Okay, so each cell go in opposite direction. This is called cytokinesis, which divides the cell into two cells. Okay. So here, cytokinesis, it is the division of the cytoplasm. Okay, don't get confused between the cytokinesis and mitosis. Mitosis is division of nucleus. Cytokinesis is the division of cytoplasm between two daughter cells, each with genetically identical cells. Cytokinesis differ in animal and plant cells. In animal cells, cleavage furrow separates two new nuclei. In plant cell, a plate or a cell plate forms. So again, I will say this part again, okay? So here in the cytokinesis, these are the cells. These are the two daughter cells. They will be broken down, okay, and give you two cells, okay? But this is happen in the animal cell. But what happened in the plant cell? The plant cell don't get separated this way, don't make a cleavage, but in the plant cell, let's imagine that these are two cells, plant cell. Here you can see a wall is formed or a cell plate is formed to separate the two cells. So now the two cells are separated. Okay, so these are the whole mitosis division. It consists of prophase, here. This is the interphase, okay? And the chromosomes start to line up in the metaphase, okay? In the anaphase, the spindle fiber becomes shorter and pull each chromatid toward itself in opposite direction. And after anaphase, telophase and cytokinesis happen. Okay, so now we have finished talking about the stages of the cell division, and we have finished talking about the first part of revision. See you next video. I will make you another video for revision 2. Thank you for listening.